Hello, my name is Tahmid Talha, and today I will be covering the research article LPAR5 regulates the CD8 T cell response to persistent virus infection by altering exhaustion programming, survival, and NK receptor expression. So here's the abstract, and although I want to go over the abstract, it's a bit too long as it is and a bit too incomprehensive as it is. So here are the three main points I want you to take away from this abstract. So the problem that is being presented in this abstract is that due to repeated use from chronic viral infections and or tumor development, CD8 T cells can become exhausted and have less functionality overall. And we'll explain a bit more about what exhaustion means in relation to CD8 T cells and what this exhaustion does. And the route to the solution that's been presented within the abstract was that previous research into LPA, known as bioactive lipid lysophosphatidic acid, shows its ability to regulate CD8 T cell functionality through the LPA receptor 5, also known as LPAR5. And so the solution has been to do further research into how LPAR5 regulates CD8 T cell functionality. And so through this research that they've done and this research paper that they've presented, they've done so. So some further background. So the state of exhaustion, as previously mentioned in CD8 T cell, is a mammalian evolutionary adaptation to prevent lethal immunopathology to the host. But in relation to cancer development and persistent viral infections, the mechanism contributes to impaired elimination by the adaptive immune system. So basically, um, the mechanism of exhaustion exists so as to prevent immunopath lethal immunopathology to the host, which would straight up kill the host. However, in relation to cancer development and persistent viral infections, which would eventually kill the host, this exhaustion uh, feature hasn't done much, or rather, this exhaustion mechanism has hindered the host's ability to fight back against viral infections and cancer development, persistent viral infections. Sorry. So current research in T cell exhaustion has three different ways about going about it, and so it either aims to well not either it aims to prevent terminally exhausted cells to exist, just not have the cells become exhausted, to reprogram terminally exhausted cells into more functional cells, essentially like repair them back or restore some amount of function, or to reverse aspects of the programming to straight up not allow them to program them uh, completely to becoming exhausted and not being able to function properly. And so, um, while normally LPA exists in low levels in mice and humans, they increase significantly in various cancers and chronic viral infections, including HIV, hepatitis C and B viruses, thus indicating some sort of relationship that LPA has with um, chronic viral infections and various cancers. And so, through this relationship, we could investigate how they interact specifically. So, nascent LPA binds to LPAR 1 through 6, which then mediates diverse biological functions across numerous cell types, some of which have been previously demonstrated by the researchers who created this paper to be impairing antigen mediated TCR signaling and release of perforin, IFNY, and IL-2 at the immune synapse. So, LPA binding to LPA receptors 1 through 6 has a pretty significant function in the way that humans or the organism can survive. And some of these functions that the LPAR receptors can enable are, as shown, antigen mediated TCR signaling and release of preferrin and so on and so forth. So the hypothesis and experiment being presented in this research paper is as follows. So before this research paper, it has not been yet investigated whether LPAR5 signaling by CD8 T cells also influences the antiviral response. So this paper aims to do that. And the researchers of this paper hypothesized that the LPAR5 mediated changes in gene expression during persistent antigen exposure regulate CD8 T cell exhaustion programming. So, LPAR5 has the ability to regulate CD8 T cell exhaustion programming, or at least that is their hypothesis. And this will occur during persistent antigen exposure, thus meaning that the researchers have decided to more so focus on viruses. And so, to expand on this, to investigate this hypothesis, the researchers used two types of lymphocytic choriomeningitis virus, CL clone 13 and Armstrong 53B, on wild type mice as well as LPAR5 deficient mice or LPAR deficient mice, which usually results in a chronic infection with persistent TCR signaling in them and creates highly specific CD8 effector T cells and long-lived memory cells. So it's easier to track um, the immune response to them, and what kind of effect that the absence of LPAR5 has on the uh, on these soon to be formed CD8 effector T cells. And so, comparatively to ARM, clone 13 targets CD8 T cells more so, like they have a stronger correlation with each other. And so, this LCMV, short for lymphocytic choriomeningitis virus, this LCMV system allows the researchers to demonstrate that LPR5 acts in a cell intrinsic manner to regulate CD8 T cell survival and the transition from regular CD8 T cells to exhausted CD8 T cells. So, this system will be able to link the relationship between LPAR5 to CD8 T cells, as well as how the CD8 T cells then survive and function or become exhausted, so on and so forth. So, here's a brief overview of the experimental methods and data. So this study, as I've mentioned previously, used wild type and LPAR deficient mice, including P14 TCR transgenic lines, to examine their respective CD8 T cell responses to the LCMV, ARM, or CL13 viruses. So the adoptive and also, the adoptive transfers of wild type and knockout P14 cells were performed before infection with the viral challenge delivered IP or IV, depending on the strain, and the viral stalks were grown in BHK21 cells and quantified by plaque assay, BHK21 cells relating to baby hamster cells. And the tissues that were gained from the mice's, the samples or of this experiment, were processed into single cell suspensions for flow cytometry, cytokine staining, apoptosis assays, proliferation assays, and survival measurements. Bulk RNA seq was performed on sorted CD45.1 and CD45.2 P14 CD8 T cells. Histology and serum liver and enzymes were assessed at day 9. Data was analyzed using standard statistical tests such as the Mann-Whitney test and the unpaired t-test, and DESEQ2 for the RNA sequencing, with results reproduced across roughly 6 independent experiments. And so here are the results that were gained from this. LPAR5 deficient mice were more likely to die to LMCV infection mediated by CD8 T cells as opposed to wild type mice, driven by the CD8 T cells, as the CD8 T cells without LPAR5 were expanded more so than CD8 T cells with LPAR5. and had eventually caused excess tissue damage and lethal immunopathology due to not being able to enter terminal exhaustion. And so this is occurring in the test without LPA present, and so or as much LPA. And so in the mixed transfer test, the same had essentially occurred with wild type CD8 T cells having a more controlled response and transitioning into terminally exhausted states with more normally preventing immunopathology. And however, unlike current viral infection findings that are present throughout the field, LPAR5 deficient CD8 T cells proliferate strongly after TCR stimulation and perform better than wild type cells in tumors, especially in the presence of LPA. So, without the TCR stimulation, the LPAR5 deficient CD8 T cells were not able to do as well. In fact, they were fighting off, or rather, they were affected by lethal immunopathology. However, after TCR stimulation, they were able to do better than even wild type cells. And so these effects were present more so during clone 13 infections as opposed to ARM infections, which where both types of mice displayed similar results. So, because of the clone 13 having a closer correlation to CD8 T cells, the better results were, or the results were more easily, uh, or rather, the changes were more easily apparent. And so, overall, as shown by the results of this experiment, LPAR5 serves as a brake that limits CD8 T cell expansion and promotes exhaustion to protect the host from immune mediated damage during chronic viral infection. And so the conclusion that could be made overall, considering all the results, was that LPAR5 regulates CD8 T cell exhaustion through modulation of the NK receptor expression specifically. So through the NK expression, receptor expression, they are able to control whether or not the CD8, the LPAR5 is able to control whether or not the CD8 T cell would become exhausted at all, not exhausted, or somewhat exhausted. And so LPAR5 signaling regulates the CD8 T cell response to persistent virus infection by altering T cell exhaustion programming, survival, and NK receptor expression. So overall, LPAR5 was regulating the CD8 T cell response as a whole. And so the mortality of LPAR5 mice in the experiment was mainly mediated by the expression of LPAR5 by CD8 T cells, although a definite mechanism between, uh, sorry, LPAR5 mice that do not have the LPAR5 were uh, were more likely to die, but a definite mechanism between why these LPAR5 deficient mice were dying hasn't been identified by the experiment. Overall, both past and current results demonstrate that LPAR5 normally restrains CD8 T cell immunity, and targeting LPAR5 could enhance both antiviral and anti tumor T cell responses, although there is still the threat of immunopathology, lethal immunopathology. But through the previously mentioned TCR stimulation, they were able to overcome this. And so, future research that could be done would be more research into LPAR5 deficient CD8 T cells, the mechanism by how the mice deficient in LPAR5 could also be studied, and research into how LPAR5 affects CD8 T cells in response to tumors. Thank you for watching.